Okay, so hello everyone. I'm Marco P. Pastrana and this is for my research methodology class. Final requirement, which is the report on my research proposal. So my research proposal is about the development of a rapid diagnostic test kit for leptospirosis using the Leptospira interrogans cerebromanilae as antigen source. So I am Mark Paul Pastrana, and my advisor is Sir Edilberto Manahan and Dr. Kulachart Jang Patarapong sa PhD from Idol University. So in order to introduce myself, I included the curriculum vitae part into the earlier slides, and I will run past through the presentation in a faster phase in order to save the space of the report video, and for me to be able to upload it better. Okay? So to start, I am Mark Paul P. Pastrana. I am a registered medical technologist. I am a certified drug analyst. I am a diagnostic medical sonographer, malaria microscopist, and phlebotomist by profession. So I am residing in Banga First, Plaridel, Bulacan. And my research interest is about laboratory management, malaria, ultrasonography, phlebotomy, toxicology, and everything under the scope of medical technology or under the topic of health. So for my education, I currently took the Bachelor of Science in Medical Technology in UST, and I have graduated in 2016. I took the boards as well and passed it on 2016 as well. So I'm undertaking my master's degree now, currently a student, and also in the same school, University of St. Thomas. And for my professional experience, I work exclusively for our laboratory in Bulacan, Pastrana Clinical Laboratory. So we specialize in diagnostic and medical tests, medicals of persons who will go to work, and diagnostic testing and consultation from our specific doctors. So why did I choose this study? This is a simple abstract of my whole research in order to give you a glimpse of what it's all about. So the purpose of this study is to make an alternative rapid diagnostic test kit utilizing the Leptospira interrogans cerebromanilae strain LT398 in order to make a more efficient, faster, specific, sensitive, and cheaper earlier detection way of detecting leptospirosis in our country. Because as we all know, the gold standard for detection of leptospirosis is still the microagglutination test. And currently, it is taking too long just for us to know the results. So I would like to make a test kit in order to detect the infection of leptospirosis faster, and it would save you more time, it would save you more money, and save you more resources in order to promote health in general. So this research is focused on the determination of the most prevalent species of Leptospira here in the Philippines, and also on the most pathogenic strain in the most infected region, and compare the effectivity of the alternative rapid test kit into our gold standard, which is the microagglutination test. So this is a glimpse of my table of contents. So pages are not yet included because there are still possible changes due to the updates of my Gantt chart because of the coronavirus outbreak. So first one is for my problem rationale. So leptospirosis is a neglected zoonotic disease that affects both man and humans. It is a bacterial infection and caused by the transmission of having the bacteria introduced on a wound of a patient. So this can be prevalent on the times where there are floods or rains, and is almost always present everywhere. Most probably and most prevalent infections are happening on those which possesses most of the samples of rats, or where rats are predominant in nature. So that's it. So just as what I have told you a while ago, we wanted to make a kit because the gold standard is taking too long, and we want to reduce the length of the turnaround time of the process of detection, and we want to develop a rapid diagnostic test kit that is specific for the detection of a specific antigen in our country. Okay? So the statement of the problem, this is the reason why I'm doing this. So the current available commercial diagnostic test kits in the Philippines is currently not designed or not specific for the Leptospira species that is prevalent in our country. As of what I know, the Philippines is utilizing a leptospirosis kit that is based or manufactured mostly in India. So I would like to pursue this research because I would like the Philippines kits to be specific in diagnosing the leptospirosis that is also specific for the Philippine infections, most likely by the strains that is present only in the Philippines and would not be detected if we use another kits which is commercially manufactured. So the problem addressed in this study is that there is an unavailability of the rapid test kit because we only use the microagglutination test, and if you use the commercially available kits, it is not specific. So we would like to also have an alternative for the gold standard that does not require so much skill. It also would not require so much effort and would not require so much supervision because the kit is semi-automated. Once you drop the serum of the patient on the kit, it will provide result in just 10 to 15 minutes. Okay? So for the objectives of our study, it's simple. We want to develop a rapid diagnostic test kit. That is our primary objective. For our secondary objectives, we need to determine the genetic profile of the prevalent Leptospira, compare our kit to the commercially available kits worldwide, and compare the sensitivity and specificity of the kit against the current gold standard, which is the microagglutination test. So what would be the significance of this study? So that is very basic. We would like to have an alternative for the gold standard. We want to reduce the turnaround time, and we want to reduce the cost per test, the effort, minimize the resources that will be consumed during the administration. However, there are certain limits to my studies. Although I would be utilizing the Leptospira strains in our country, I cannot use every single strain of Leptospira in the Philippines. I would only focus on the most pathogenic and most prevalent strain of leptospirosis here in the country. Okay? So for the review of my related literature, I will show you some of the most important studies that I have incorporated in my research. They served as the mother articles in which I have based my study and in which I stand with against and I stand with on my study, not against. Just joking. So for the first one, it is authored by Villanueva, taken from the American Journal of Tropical Medicine and Hygiene. The title of the article is The Serologic and Molecular Studies of Leptospira and Leptospirosis Among Rats in the Philippines. Okay? So what is the major data and result? 92% of the rat serum samples were positive for anti-Leptospira antibodies. So they have been making anti-Leptospira antibodies in order to counter leptospirosis. And in that way, they have been able to determine the most prevalent strain, which is the LT398, which is the Leptospira interrogans serovarmanilae. Okay? That would be our utilized antigen in order to make our rapid diagnostic test kit. So the problem is that the study only tested samples and Leptospira predominance in carrier population. So what specific objectives will this answer? So the specific objective number 1. So we will determine the genetic profile of the most prevalent Leptospira in the Philippines. So because this study showed us what is the most prevalent strain, that would be our focus for this study. Okay, so the next one is the clinical evaluation of rapid diagnostic test kit using the polysaccharide as a genus-specific diagnostic antigen for leptospirosis in Korea, Bulgaria, and Argentina by Lee et al. Okay, so the major data and result is that the rapid test kit evaluation resulted in a test performance with 94% sensitivity and 98% specificity. So this study is the one which I have based my comparison with, because they have been able to compare the kit that they did into other commercially available kits. So when we make our kit from the Philippines, this would be my study that I would use in order to compare it also within the kits of other commercially available kits worldwide. So the only problem is that although they have given the result and the effect or the specs of the kits, the specificity or the sensitivity, it only focuses on Korea, Bulgaria, and Argentina, and does not involve any other country. So for the third one, evaluation of a rapid kit for the detection of IgM against Leptospira. This one also showed us the negative predictive values, the positive predictive values, sensitivity, and specificity. So this is where I will base my statistics when I am about to evaluate the kit that I will do in order to compare it with the gold standard, its effectivity, and commercially available kits. So the gaps is that the kit is not able to detect the early or acute infection of leptospirosis. And then the specific objective that it answers is that the kit that we made would be compared into a gold standard, which is the microagglutination test. So when we compare the kit to a gold standard, that is when we will apply the statistics here. Okay, so next is that the human and animal leptospirosis in Pacific Islands and clinical epidemiological status of leptospirosis in a tropical and Caribbean area of Colombia. So the major data of these two studies is that leptospirosis prevalence is dependent on an area, and reservoir and carriers play an important role in the disease. And then microscopic agglutination test is usually positive 10 to 12 days after the first onset of symptoms. So even if you have leptospirosis, in order to detect it, microagglutination test would take almost 2 weeks just to detect it. And then this answers the specific objective number 1, determine the genetic profile, and the main objective, which is to create a rapid diagnostic test kit that would detect leptospirosis. The only problem is that with this study, Philippines is not part of the Pacific Islands, and that their study focused more on the treatment instead of prevention and early detection. So for the chapter 3, for the research design, I utilized an experimental research design and develop rapid diagnostic test kit using the LT398 and tested against the gold standard, which is MAT. So here we will utilize the multi-gene targeted real-time polymerase chain reaction to diagnose the strain. And after the culture, we'll either get it from UPLB, University of Philippines Los Baños National Collection of Microorganisms, or the Philippine General Hospital Leptolab, or the University of Santo Tomas Collection of Microbial Strains. And then after we get the culture, this will be preserved because this will be the source of our antigen. So in order to isolate our sample, um, the culture that we've got will extract and isolate, and from there we will determine the serovariations of the Leptospira. Okay, so for the sample serotyping, a serotyping procedure will be performed to detect the serovariant, and then microscopic agglutination test will be utilized with a serovariant specific anti-sera panel from rabbit, which covers 24 Leptospira serovariants. So this procedure will be used using microtiter plates, serial two-fold dilutions of rabbit anti-serum, 2 hours of incubation, and then check with dark microscopy in order to detect agglutination of bacteria. So the presumptive isolates will be determined when results from both molecular typing and serovariant determination is performed. So when we've got their serotype and their isolate, we will then culture and reproduce our sample. The bacterial culture will be performed on the Central Lab of University of Santo Tomas and involves the aseptically removed rat kidneys. Then, our culture media will be 5-fluorouracil containing modified Korthoff's medium supplemented with 5-fluorouracil containing Ellingshausen, McCullough, Johnson Harris or AMJH medium and 10% rabbit serum. So Leptospira sample or culture will be preserved via cryopreservation or deep freeze method in order to prolong its life and to be able to use it as we transport it into Thailand for the rapid test kit and kit manufacturing. So from a fresh culture isolate, we'll get a liquid culture that will be directly combined with an equal amount of glycerol. All of the possible contaminants will be washed, and then we will also not resort into the repeated freeze thaw method in order to preserve the integrity of our sample. As of what I've stated a while ago, in order to determine the genetic makeup of our sample, multi-gene targeted PCR will be used. And then the DNA will be extracted from the culture isolates as a sample for the RT-PCR test. And then for the rapid diagnostic test kit production, the antigen that was isolated will be utilized as a marker for the developed rapid diagnostic test kit. The specific antigen will be placed in an immunochromatographic test column. This test kit will determine the IgM antibodies present in the serum of the patients, and this test will utilize nano gold particles as agglutination revealing marker. So the rapid diagnostic test kit, once produced, will be now compared to the microscopic agglutination test. So in order to determine its efficacy, it will be compared to the gold standard. So the comparison involves the testing of predetermined positive and negative leptospirosis samples alongside MAT and the rapid diagnostic test kit. So all of their tests will be done twice, one from the rapid diagnostic test kit and one from the MAT in order to compare their effectivity. And there, if you see this in a zoomed image, this would be my budgetary requirements, and all of them would be amounting to a total of 413,800. However, when it comes to my Gantt chart and proposed Gantt chart, me and my advisor have come to conclude that it is not yet a proper time to set a specific time goal or time limit within this study because of the outbreak of coronavirus and because the quarantine is still present. We cannot determine yet the the sequence of our study, or how will we approach the study in a more safe manner. But rest is assured that once the quarantine is lifted and the international travels are now safer, we will proceed as quickly as possible with the study. And that concludes my research presentation. I hope I have made you more enlightened with my research, and I also hope that you consider making other kits as well, for not only leptospirosis but maybe for coronavirus as well, and etc. Thank you and God bless.